Hi, welcome to my presentation of our work of benchmarking deep learning methods dedicated to RNA binding protein binding preferences. So, first, a bit of context for this work. There's been many machine learning methods that have been developed to learn from clip-based data, learn the binding preferences of RNA binding proteins, what are the sequence determinants that are recognized by these RBPs. And the task for these models is to then be able to predict on unseen sequences, such as transcripts that were not expressed in the cell type used by the experiments. And in recent years, there's been many deep learning methods that have been developed for this task, with various choices of architectures. But the issue is that from one publication to another, you have many variations in what datasets are used, how the data is sampled, what are the methods that are compared for their own benchmark. So overall, it makes it very difficult to identify what are the critical choices that one can make in terms of architectures or input modalities. So to address that, we established a benchmark where we evaluated 10 deep learning methods, along with one shallow learning method for reference, on data sets, on 3 data sets, providing us with positive control examples. And so for the classification task and the negative controls, what we did was to establish 2 strategies of sampling. The first one, we labeled -1, consists in classifying between regions bound by an RBP of interest versus regions that were not bound by any RBP in the data set. The second approach, which we label -2, consists in the classification between regions for an RBP of interest versus regions bound by other RBPs. And this setting is critical because CLIP is known to be associated with specific sequence biases that the models might learn from in the -1 setting, while in the -2 setting, these biases will be equally present in both classes. Now, going directly with the results, we measured performance of the different methods through the area under the rock curves, here presented for the -1 setting across the 3 data sets. So each method is represented by a box plot representing the performance across the different RBPs within this data set. And the first thing that you can see is that the graph plot method, the shallow learning method, is in gray, is outperformed by all the deep learning methods across the 3 data sets. Coming to the deep learning methods, we observed a range going from the best performing model, namely RNA-prot, recurrent neural network model, down to DeepRAM, another recurrent neural network, with the second best being Pyster, a CNN-based model. So the first thing we observed already is that there was no clear advantage of one architecture over the other. And this was confirmed in the -2 setting, which is presented here, where you can see nonetheless that there's an overall, even though the ranking between the different methods remain the same, there's an overall degradation performance owing to the fact that the classification task is more difficult there. This -2 setting was also the occasion to compare the performance of the multi-label models, because these learn from each dataset, they learn at once the binding preferences from all the RBPs, but they cannot learn between bound and unbound regions. And so what we observed was that with the exception of multi-RBP, the multi-label models outperform the single label models. And what's nice is that they can be easily adapted for classification of unbound regions, yes. Going into the investigation of specific models now. RNA-prot was seen to be the best performing model in both -1 and -2 setting, but had the greatest decrease in performance from -1 to -2. So one thing that was striking is that this model was using, compared to the other, was this model was using not only the sequence of the bound or unbound regions, but also the sequence evolutionary conservation and genomic annotations such as intron or exon. So there, what we did was to train new RNA plot models that exploited only the sequence. And what you can see is that there's a great outperformance of the full model compared to the sequence-only models, both in the -1 setting and to a lesser degree in the -2 setting. Another input modality that we explored was the contribution of secondary structure, so specific conformations that are adopted by RNA sequences, which can be either predicted from the primary sequence or measured with various techniques. And what we did was to compare here the performance of PrismNet, a CNN model that considered both the sequence and the in vivo measured structure, against the performance of models that exploited only the primary sequence. And what we saw is that first, there's only a few RVPs that seem to benefit from the additional modality. But the striking observation is that this is true in the -1 setting, but then if you follow these models in the -2 setting for the classification between regions bound by different RVPs, the advantage from the secondary structure disappears, suggesting that only few RVPs actually recognize specific RNA structures. Last but not least, we addressed the question of generalization of these models by exploiting the ANCHOR dataset, where we had 59 RVPs that were measured in 2 cell types, namely FG2 and K562. And yes, the first thing that you can see on the scatter plot on the top is that the models showed comparable performance from one cell type to another. But then, if you apply models that were trained on one cell type on the validation data from the other cell type, what you can see in the scatter plots at the bottom is that there's a general degradation in performance that is mostly pronounced for the low performance models, suggesting that you have to be cautious in the generalization of poor performing models. I will stop here for the sake of time. Thank you for watching this video. Please come to my session on the Wednesday afternoon, where I will be talking during the iRNA track. Otherwise, I will be at poster C337 if you have questions. Thanks.